Hi everyone, welcome back to Teacher Kayal's Guide to Biology A+. In this video, I'll guide you through answering SPM trial questions on the topic movement of substances across the plasma membrane, focusing on osmosis. This diagram shows a selectively permeable membrane separating two solutions containing water molecules and sucrose molecules. As I have explained in my previous video, selectively permeable membrane is permeable to water molecules but not permeable to sucrose molecules. Imagine you are in a crowded area that makes you feel pressured. Naturally, you will want to move out from the crowded area. Water behaves the same from a region of higher water potential to a region of lower water potential. D means remove, saline means salt, Asian means process. So, desalination means the process of removing salt. In this process, pressure is applied to push seawater through a selectively permeable membrane. The selectively permeable membrane allows water molecules to pass through it but prevents foreign particles such as salt and microorganisms. As a result, water is forced to move against the water potential gradient from a region of lower water potential to a region of higher water potential, which is the reverse of process of osmosis. For a 2-mark question, write the top 2 points. For a 3-mark question, you need to write the extra point. When a cell is placed in a solution, the solution is called isotonic solution if the solution has the same amount of solid molecules as the cell. The solution is called hypotonic solution if the solution has less solid molecules and more water molecules than the cell. The solution is called hypertonic solution if the solution has more solid molecules and less water molecules than the cell. When a red blood cell is immersed in a hypotonic solution, water molecules diffuse into the red blood cell by osmosis. The red blood cell bursts and undergo hemolysis. When a red blood cell is immersed in hypertonic solution, water molecules diffuse out from the red blood cell by osmosis. The red blood cells shrink and undergo radiation. If the diagram states the solute name, your answer must be specific to the solute name. Don't generally answer region with high concentration of solute without stating the solute name. Concentrated salt solution is hypertonic solution compared to plant cell because concentrated salt solution has more solid molecules and less water molecules than plant cell. So, water molecules diffuse out of the cell by osmosis. Consequently, the vacuole shrink, causing the plasma membrane is pulled away from the cell wall. This creates an empty space between the plasma membrane and the cell wall because cell wall doesn't restrict any substances. The concentrated salt solution from outside enter the cell and fill the space. A hypertonic solution causes water to diffuse out of the plant cell by osmosis, causing the plant cell become plasmolysed. You only need to label the affected cell components, which are the vacuole and the plasma membrane. To restore the plasmolysed plant cell to normal condition, we need to immerse the cell in a hypotonic solution compared to the cell. So, water molecules will diffuse into the cell by osmosis. Consequently, the vacuole becomes bigger, causing the plasma membrane is pushed to the cell wall. To restore the turgid plant cell back to the plasmolysed condition, we need to immerse the cell in a hypertonic solution compared to the cell. So, water molecules will diffuse out of the cell by osmosis. Consequently, the vacuole shrinks, causing the plasma membrane is pulled away from the cell wall. The previous questions involved a single cell, where the condition is described as turgid or plasmolysed. In contrast, this question involves a plant strip that consists of many cells. The inner surface of the mustard green strip has cells that normally allow water movement by osmosis, whereas the outer surface has a rigid epidermal layer that resists water movement. When a plant strip is immersed in a hypotonic solution, water diffuses into the inner cells by osmosis, causing them to become turgid. Turgid cells are slightly longer than normal cells. So, when many adjacent cells are turgid, the inner surface becomes noticeably longer. Since the outer epidermal layer remains the same length, the longer inner surface causes the strip to bend and curve outwards. When a plant strip is immersed in a hypertonic solution, water diffuses out of the inner cells by osmosis, causing them to become plasmolysed. Plasmolysed cells are slightly shorter than normal cells. So, when many adjacent cells are plasmolysed, the inner surface becomes noticeably shorter. Since the outer epidermal layer remains the same length, the shorter inner surface causes the strip to bend and curve inward. The concept of this chili strip experiment is the same as the earlier mustard green strip experiment. We have learned the effects of different types of solutions on a single plant cell and on a plant strip. Now, let us learn the effect on a whole plant. When too much fertilizer is added to the soil, the high solid concentration makes the soil become hypertonic to the plant cell. Water molecules diffuse out of the plant cells by osmosis. When many cells throughout the plant become plasmolysed, the plant will wilt. This diagram shows the process of making kimchi. Particularly, the first step is to preserve cabbage. Cabbage is immersed in a concentrated salt solution because concentrated salt solution is a hypertonic solution. So water diffuses out of the cabbage cells by osmosis, causing the cabbage cells to become dehydrated. As we know, microorganisms need water for survival. So when there is no water, microorganisms cannot live on the cabbage. Therefore, the preserved cabbage can last longer. The second and third steps are mainly to enhance the taste and flavor of the kimchi. Similar to cabbage preservation, mango too can be preserved. The choice of using a salt solution or a sugar solution depends on the type of food and the flavor desired. Salt is commonly used for savory food like cabbage, while sugar is used for sweet food like fruits. Isotonic drink help to recover loss of water and electrolytes rapidly because it contains similar concentration of salt and sugar as in human body. Salt contains electrolytes such as sodium, while sugar contains glucose. The glucose helps sodium to be absorbed from small intestine into blood plasma faster than usual. The sodium in blood plasma then will reduce the water potential of the blood plasma, so water molecules from small intestine will diffuse into blood plasma by osmosis. Similar to isotonic drinks, saline solution helps in rapid rehydration. Saline solution is usually given to patients admitted in hospital. Similar to isotonic drinks and saline solution, ORS solution helps in rapid rehydration but usually taken when recovering at home, not in hospital. For the point number 3, take individual if the person is not treated by doctor yet. If the person already been treated or seen doctor, then you have to state it as patient. This question is almost the same with the previously discussed question, but more marks is allocated. So you have to explain more detailed. That's all for this video and I'll see you in the next video. Remember to study smarter, not harder.